Okay, well, today I'm going to clean up the loose ends. First off, so I'm going to touch on a number of things. Any of which, well, a couple of which at least, could be, again, an entire course. So one of the things I'm going to talk about is information processing in the brain. We've talked about sensory and motor function, but the vast majority of neurons actually are not either sensory or motor neurons. The bulk of them are these interneurons, and I'm just going to give you the very roughest overview, in no way in depth on that functioning. Related to that, I'm going to talk, I think, one slide about memory. You could certainly devote your entire graduate career to the study of that. But you'll get one slide out of me. This isn't a neuroscience class. And then I'm going to talk about reflexes. Just a little bit of how reflexes work in the body. So those are our topics for today. Friday, not tomorrow, Friday, you guys are going to meet with me individually as groups. But I should have made this point clear earlier on. I'm going to give a quiz over this material. You can take that quiz at any point during the hour of class time when I'm not meeting with you. It's going to cover all of this material on the central nervous system and peripheral nervous system. So these 5 lectures, okay? And then we'll start next week with eye, and then ear, and then we'll have a midterm, and then you guys are heading to Aruba or wherever you're going. Take me with you. Okay, let's jump into it. Right. So there are approximately 10 million sensory neurons in your body. 10 million points of contact with the outside world. There are far fewer motor neurons. There are about 500,000 motor neurons, half of a million, which is how your mind exerts control over the body. All right? So those numbers are dwarfed by 3 or 4 orders of magnitude by the number of interneurons. Okay? Approximately 20 billion interneurons in the central nervous system, who are responsible for integrating all of that information collected by those afferent neurons, those 10 billion points of contact with the external world, and integrating, processing, analyzing, and then exerting executive control. These interneurons are grouped into neuronal pools. Neuronal pools are collections of interneurons that have discrete entry and exit points. Okay? There may be a vast array of neurons, interneurons in particular, in these neuronal pools, but they're going to have specific points of entry and exit. And each of these neuronal pools is associated with a particular function. It can be a neuronal pool related to a learned function such as playing the piano or walking, or the rhythmic coordination of rhythmic breathing. Each of those functions is going to have a neuronal pool that guides and coordinates those activities. All right. So each of the executive functions that your mind is able to enjoy is guided by a neuronal pool. So here is just a very rough diagram of what it may look like, and this is meant to illustrate this point. So there's the discrete point of input into a neuronal pool, right? And there's whatever complexity of neurons, and I was saying there's millions of interneurons within a neuronal pool. So this can have a significant complexity here, right? And then there's going to be a discrete discharge zone where the output is going to be coming from specific output terminals. There are collateral neurons associated with this neuronal pool that aren't actually discharging. They're not signaling. There's not going to be an output. But on that fringe, they're getting facilitated. So though the membrane potential, for example, if we want to talk about it in terms of that, that membrane potential, we'll say the threshold for firing here is at -60. Resting is at -70. These neurons are all hitting threshold and discharging. These are maybe at 65. So if they're getting input from a parallel neuronal pool that's overlapping in this facilitated zone, then we're going to have discharge through these regions that have been facilitated. Right? So along the margins of a neuronal pool, there are these facilitated groups of neurons. All right, sort of like the fuzzy boundary of the neuronal pools. Is that making some sort of sense? Yeah. So I mean, the wiring in there is a unique wiring pattern to the brain, right? But it is dynamic. And, a unique pattern at any one point in time, but it's dynamic. It shifts, it changes, and there is overlap between discrete regions of the circuitry for in search of a different term than neuronal pool, but meaning that. Okay, so let's look at some of the types of, yeah, Jacob.
Oh, so does the amount of those peripheral neurons that are activated, does that have anything to do with, say, how hard you're thinking about doing the particular thing, or is there not really a correlation?
This is talking in such broad generalities that I can't really answer that. Okay. It could be due to intensity of stimulus or collateral stimulus. Maybe you feel something on your arm, and then you see something on your arm, so you're getting a parallel bit of stimulus to the motor cortex or something. It's hard to answer that question. Yeah, the intensity with which you're thinking is hard to quantify. Yeah.
I don't think I quite understand. So the ones in the middle have more synapse?
Well, yeah, there's a lot of synapse on them. So these are going to, and they're at the core of this neuronal pool, right? So when there's some sort of stimulus coming from this input neuron is having a action potential. It's firing on all of these. These neurons are hitting threshold and firing, so they're discharging. This is the output. These ones on the periphery, they're getting facilitated. They're getting pushed towards threshold, but they're not reaching threshold. So they need some other collateral input from some other neuronal pool that is overlapping here with some other synapses that may not be firing. The input to that collateral neuronal pool may not be getting stimulated. Yeah.
And these exist only in the brain, or
this is in the central nervous system that we're talking about. These are interneurons. Yeah, you missed the first minute or two of what I was talking about. But
so those are also in the spinal cord.
Yeah, you're going to have neuronal pools in the spinal cord, but they tend to be in higher structures. The more sophisticated neural pools in the higher structures. So let's look at I don't know if any of you remember circuitry from physics in high school, or if any of you took it here in college, but the brain certainly has its own types of circuitry, and we're going to see those in the next couple slides. So there's divergence. So this is where you have a discrete, limited input from, well, just in this picture, a single neuron is getting stimulated somehow from a sensory neuron, and then it's synapsing on multiple downstream elements. All right, so you can have multiple neurons, and that can have a further arborization. This is a mechanism where a signal is amplified. Say it's recognized that whatever stimulus is coming in is important, and so we're going to amplify that signal so that we can distribute it across multiple neuronal pools or brain regions, whatever. There is also convergence. So say each of these neurons here is receiving discrete output from different neuronal pools, but the consequence is that there needs to be a unique output. We have convergence impinging upon a single neuronal output there. So there's just sort of the opposite of divergence. Serial processing. This is simply a case where you're passing the buck in a linear sense. You're not amplifying a signal or converging a signal. Just neurons in serial. There's also parallel processing, where you have two signals which have received identical input and are processing information side by side simultaneously. Okay. And then there's reverberation. So reverberation is a positive, it can be a positive or it can be a negative feedback mechanism. This slide says positive, I shouldn't say that, it's just a feedback mechanism. Where you're getting some sort of input, it synapses on the downstream element, which continues to propagate that signal into whatever other downstream elements there are, but there's a collateral that goes back and is somehow going to modify that incoming signal. It can amplify it. So here's the general pattern. It can go back and modify it, thus strengthening that signal or mimicking the initial signal that came in. All right. That postsynaptic, that synaptic terminal here can be inhibitory, and then this can be inhibitory as well, thus dampening this inhibition here. All right, so you have a signal that is going to dampen activity in the downstream elements, but the reverberation lessens the effect of that, so immediately attenuates that. And then we can have a mixed response where this is a potentiation, an excitatory postsynaptic potential here at this synapse, but then this is inhibitory, which is going to also dampen the output of that downstream neuron. Oh, this was meant to show that you can have it such that a collateral from its own neuron, from the neuron, is actually attenuating itself. So there's an internal mechanism. I forgot that there wasn't a neuron in there. Okay, so there's a lot of different permutations, and it's hard for me to give you examples of them because they're found throughout the nervous system. All right, so obviously the brain is an enormously complex system. And it's not just complex in terms of the linear connectivity, which it is. I told you there were 20 billion neurons. Holy smokes. And those are just nodes, right? 20 billion nodes in a network. And they're all not lined up in a linear chain, so there must be a far greater synaptic connectivity because multiple neurons are connecting to multiple other neurons, right? But let's take it a step further, where it's not all neurons connect to each other in the same way. There can be spatial variation in how a neuron is synapsing on another. So here we have some upstream neuron. It's going to synapse on the body of that downstream neuron, and we have, the chain being passed. Here we can have an axosomatic synapse. So the axon is synapsing on the soma closer to the axon hillock, is going to have a greater effect on the ability to reach potential at that axon hillock. We have an axodendritic synapse further away, more distal to the axon hillock. So this is going to have a higher frequency of stimulation to have the same effect as this axosomatic synapse, assuming they're both excitatory. All right. Furthermore, there's axoaxonic synapses, where a neuron can get in there and immediately, right before there is an action potential, it can attenuate that action potential. It can attenuate the action potential. Say an action potential is coming along here, but we want to stop it. You can do that with an axoaxonic synapse. So where on any of these, where the synapse exists on the body or processes of an axon are going to have sort of an informational content, and they're going to be able to modify the functioning of that circuit. And it gets much more complicated than even this. So, for example, on dendrites, there are these dendritic spines. It's something I talked about I would have talked about in a previous lecture that I cut out, but we're gonna pass it by. It gets quite significantly sophisticated. So I used this term already: excitatory postsynaptic potential and the inhibitory postsynaptic potential. So excitatory is just going to be a depolarization. You should already have been able to guess that. You know that. So that is taking the resting potential from -70 towards threshold at -60. If there is a synapse that's causing an inhibitory postsynaptic potential, right? So opening up a potassium channel, for example, driving it towards potassium's equilibrium potential of -90, that's going to be a hyperpolarization. That's inhibitory. That's making it harder to reach threshold. Okay? So you can have synapses that are going to push you to threshold or ones that take you back from the brink. And so here we go. Here, for example, maybe this is a nicotinic synapse, and it's got acetylcholine, and we have this depolarization where we push the membrane potential towards threshold, and then the stimulus is removed, and you come back down here. It wasn't enough to hit action potential, right? So then second stimulus, we're going to have a GABAergic receptor, maybe, and it's going to open a chloride channel, or maybe some potassium channel gets opened or something. It's going to push this membrane potential towards -90 if it's potassium, for example, this is an inhibitory potential. It's getting us further away from this threshold. Getting further away, we move the stimulus, etc. Maybe we apply both at the same time from two collateral, from two parallel inputs, one inhibitory, one excitatory. Membrane potential doesn't go anywhere. So as you have an excitatory postsynaptic potential, this is edging you closer to that threshold. That is a potentiated neuron, meaning now that neuron, it doesn't take a whole lot of stimulus to activate it, right? And this is like, for example, if you look at fMRI, which is not looking at membrane potential, it's looking at blood flow to the brain, but it's a proxy for what I'm going to talk about. If you look at one of those scans of a person who is thinking about doing something, I'm thinking about scratching my nose, but I'm going to wait for a light to come on before I do it, right? This could be a test maybe that they're doing in one of these tubes. I'm thinking about scratching my nose, but I haven't done it. As you're thinking about doing that, those neurons that are controlling the muscles that do this are getting potentiated. They're getting potentiated. They're getting some input that's bringing those neurons closer to potential, so that when you get that signal, it doesn't take as much input to reach threshold and have that particular action. And you're all familiar with this. Whenever the start of a race, you're waiting for the gun to go off. Those neurons in your legs and wherever are that control the muscles of the legs have been potentiated. All right. So we did spatial summation. Here's a better picture of spatial summation. Well, I guess I didn't talk about spatial summation quite. I talked about proximal and distal. Here is two neurons that arrive simultaneously. If we had one, perhaps it would not be enough to reach threshold, but because there are two that are spatially proximal, they're able to sum and reach potential, and we have an action potential here. There's temporal summation. Here's there's a single neuron that stimulates, but it's not enough to get us to threshold at the axon hillock. It's not quite enough. But we increase the frequency. Remember I talked about all the different ways, the 4 different ways that neurons encode information. The frequency is increased here, so we're able to reach threshold and have an action potential. So there's temporal summation and spatial summation, different ways of getting a neuron to fire. And here's just another diagram, quite similar to one I think I showed you a few weeks ago. Yeah.
What were the other 2 methods of?
There was modality, the labeled line code. There was duration. There was intensity, and what was it? No, I said duration. I don't know.
Intensity is, or I guess that's something that's determined at the synapse, because the action potential is always the same.
Well, there's a number of different ways that you can interpret intensity. It can be, the frequency of stimulation, I guess, is the same as intensity. You can look back at what the 4th is. I don't remember it. I want to keep thinking here. I'm sorry, I can't.
Wasn't it location?
Oh yeah, location. Thank you. That's it. Good job, Jack. Thanks for saving me. All right. So, is there anything unique on this slide that I want to say? Right. So, we have this particular output, this pattern of output. This pattern of output is being elicited either by the temporal summation, an increased frequency of this input element, or the spatial summation of multiple inputs not firing as rapidly, but in totality, giving us the same sort of output pattern, right? So this is a convergent architecture that we're seeing this pattern in here. All right. So when you look at this, we have stimulus and we get a bump, right? Where there's an excitatory postsynaptic potential. Get another one. We get another bump. We're getting pushed. The axon, that excitable membrane, is getting pushed towards minus 60, et cetera, et cetera, et cetera. Finally, we reach that threshold, boom, action potential. We go through the whole rigmarole, refractory period, repolarization, sodium, potassium, ATPase, blah, blah, blah. So I have on the question here, is this spatial or temporal summation? Can we tell? What do you think here? Yeah.
Looks temporal because there's that little stepped line.
Yeah, we're looking at time, aren't we? Yeah, we're looking at time. It's hard to tell. It's hard to tell whether there is a spatial component. Is this always the same neuron that's getting stimulated here? This diagram's not showing us that. Is there more than one neuron at each time? All right. And so I think this is as sophisticated as I'm going to get with it. No, I might have another couple slides. I talked about axo-axonic synapses, all right, and here they are. So we can have them either inhibitory or excitatory, facilitative. Here's the primary action potential, all right? And maybe this action potential is just fine. On its own, it can cause a synapse or a strong graded potential on the downstream neuron. All right, so it comes in. However, there is this inhibitory axo-axonic neuron coming here. Some other collateral neuron in green whose job it is to attenuate this. Maybe this is a constitutive neuron. It's firing repeatedly for some reason, automatically, for whatever reason. Maybe, gosh, I don't know, maybe it's dictating breathing or heart rate or something that you don't have volitional control over, right? But there's ways of inhibiting this right on site. So we have these GABAergic receptors here that are going to fire and inhibit these calcium channels. Calcium, we didn't go through this in great detail on the presynaptic side, but it's these calcium channels, you'll remember, that are responsible for the degranulation so these are voltage-gated calcium channels. The calcium floods in and then potentiates exocytosis of the neurotransmitter. But these GABAergic receptors are going to inhibit that. Maybe these are chloride channels or whatever. It's going to reduce the membrane depolarization, and it's going to reduce the electrochemical gradient that the calcium is feeling, and thus less calcium is going to enter, and less neurotransmitter is going to be released. You'll have some neurotransmitter released. You're just weakening the strength of this synapse. The point here is we can dynamically change without breaking this neuron and moving it somewhere else on the downstream neuron. You can change the strength of this synapse in situ. We can weaken it with these GABAergic receptors. Here we have catecholamine. We've got serotonin that comes in and activates more calcium channels. So more calcium floods in, and we're going to have a stronger degranulation of neurotransmitter. All right, more neurotransmitter, whatever this is. I don't know. This doesn't tell me what kind of receptors those are, what kind of synapse that is. Doesn't matter. You guys understand that? It's pretty straightforward. Anyone have a question? Lost? Confused? Excited? Ecstatic? Okay. Neural coding. So, and this is probably something that I've already kind of impressed upon you, but it's just a different way of representing it, maybe. Here we have some pressure on the skin, and we're recording the trace of a sensory neuron. We put a 2-gram weight pressure on the skin. And so we're going to have boom, 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 this kind of pattern. We increase that pressure, and we're increasing the frequency of firing of that afferent neuron, to the point where we're pushing into the refractory period of the preceding action potential here, right? We can't really increase this frequency any longer. I mean, I don't know, but you know, that's for the hypothetical here. All right. So what happens when you have an injury to a nerve? So I like to, when I can, talk about the people that were involved in this. By the way, Golgi, I had to satisfy my curiosity. He was an Italian neuroscientist of the early 20th century, and it just lent his name to the. He developed Golgi stain, which is why the Golgi body is called the Golgi body. But he first identified those Golgi spindle apparatus, which we'll talk about later. Anyways, we talk about all these guys. I don't really get a chance to talk about very many women, which is sad to me. Perhaps I should make a greater effort to comb through them. I don't know whether that be artificial or not. This woman is incredible, though. Rita Levi Montalcini. In the 1950s, she identified nerve growth factor. Look at her. They are so happy about receiving the Nobel Prize in the mid-'80s. Nerve growth factor is a peptide hormone that, you know, we have a neuron, it gets severed here. There begins to be a little bit of Schwann cell degradation. There's these degradation remnants that are left around the former nerve path, and but this nerve growth factor has a couple functions. First of all, it prevents this cell from dying. It inhibits various apoptotic pathways, and it enables this axonal stump that is left here to put out a whole bunch of rootlets. Okay, puts out all kinds of little processes, and the one that comes in contact with the Schwann tube, the Schwann cells form a tube and help guide the appropriate rootlet towards the former neuromuscular junction. In the meantime, while this is happening, that muscle cell begins to atrophy. It begins to shrink and die back. However, with time, when that peripheral neuron is able to reach the motor end plate and re-innervate that muscle, it brings muscle tonus back to where it was. Yeah?
I'm guessing if you're damaging one axon like that, you're probably damaging multiple axons in a nerve. So how does each neuron find its right hole in the Schwann tube?
Yeah, sometimes it doesn't. Sometimes it doesn't, and that's called a neuroma, and those are very painful little balls of nerve endings that can, say you cut a nerve in your hand, you cut yourself badly, and it can be a very painful little nodule that will form at the site of the inappropriately healed peripheral nerve. They don't always heal well, but the way that when they do, it's because they're following the Schwann course that was already there. Okay, so there is this pathway of Schwann cells that I don't know. I wish I could give you a better answer, because really what you're asking is how are the Schwann cells coordinating with one another to make sure that path remains integral. And I can't actually answer that for you. I've never looked into that very closely. But I can tell you that it is the job of the Schwann cells to lay down that track for the growing axonal stump to follow and reach its innervation target. So, all right, memory. So, man, there's a lot you can talk about with memory. But there's, we're going to break it up. This diagram breaks it up into short and long term. There's actually immediate memory versus short term. Immediate memory is really the function of these reverberating neurons, so that like when you're reading a book or listening to me talk, the sensory input of me saying the word hello enters your brain, and then for you to be able to put that word into the context of the longer sentence, that those neuronal pathways that registered the word hello are reverberating just for a brief period of time, so that there's some sort of temporal binding to your experience, right? So our experience is not this like chopped up into an infinitude of isolated moments, but there is some sort of continuity to your experience from moment to moment. So that's immediate memory, and that's the result of these reverberating neurons. And then you move to short-term memory and short-term storage. This is, my number is 947-863-7271, right? Call me anytime you have a problem. And you have to commit that to memory and write it down, right? That is not this immediate memory that you will encode that on short-term memory, but again, using reverberative feedback pathways. Short-term memory then gets stored to long-term memory by various structures in the brain, and there are two broad categories, one of which is broken up itself. So there's declarative and procedural memory. Declarative memory are events that you can put into words. So a declarative memory is something that is using symbolic logic, symbolic representation. All right, the sound hello means it's a greeting, and you can describe it. Or I'm doing jumping jacks, and you're watching me do jumping jacks, and you just can describe the experience of watching me do jumping jacks. All right, so the experience of watching me do jumping jacks, that would be an episodic memory. You remember that time when your crazy physiology professor did jumping jacks in front of the class. And semantic memory would be the memory, the understanding, the symbology, the semiotics of the word hello, for example, or the word camping, or whatever it happens to be, right? So that's symbolic representation. That's all declarative memory. And then procedural memory is the memory that comes with learning to play a piano, or riding a bike, or writing your name, or the muscle memory, the procedural memory of how to do something. Okay, You can put it, it may have an episodic or semantic component to it, but there's a procedural memory that dictates how some sort of efferent output, some sort of motor output is going to be used. So that's memory. And that's all I'm going to have to say about it. Yeah?
Are we not going to go over the neural reflex stuff? That was also
I'm trying.
okay.
Yeah, those were a couple. I know. I went over my cornflakes. I thought the lecture was a little bit thin, so I put some memory in there. But yeah, if I can flash through Alzheimer's, we'll get to reflex right now. Alzheimer's. Does anybody have a family member that's got Alzheimer's? Yeah, not fun, is it? No. 100,000 people die a year of it. That's 11% of the population over 65, and roughly half of people by the age of 85 get Alzheimer's. Whoa. And there's a good chance that a lot of you are going to make it to 85. Roll the dice. So what happens? You lose your memory loss for recent events. Often people with Alzheimer's remember like their childhood in vivid detail. The early memories are stored. Become moody, combative. They lose the ability in extreme cases to talk, walk, or eat. It's really heartbreaking to see. So these people, they're acetylcholine, they're acetylcholine deficient and nerve growth factor deficient. When you look at the brain of someone with dementia, you see, this is on autopsy, postmortem, you see that there's a reduction in size of the gyruses and the sulci are larger, so there's a reduction in mass of brain tissue. Yeah, that's due to the formation of these neurons that have neurofibrillary tangles, which then grow into these plaques, these senile plaques, as they're called. So this is some histology here. That's due to the formation of this beta amyloid protein that comes from the breakdown of the plasma membrane. They haven't had a breakthrough yet in the treatment of this. They try to slow it down by giving nerve growth factor or cholinesterase inhibitors to try to have the acetylcholine reside in the neural cleft longer. But these treatments all come with significant risks. So, Alzheimer's disease. There's a pretty interesting story about treating these patients by having them listen to music that they enjoyed when they were young, and it helps bring them out of the sort of stupor that's induced by the senility. Yeah, dementia.
Yeah, I've seen a lot of these studies, and they've kind of struggled with proving causality of a lot of things. We don't know whether the people are doing these things, like able to do these things, like go on Facebook and enjoy the music they like because they are not having the Alzheimer's, or that those things are actually reducing the Alzheimer's.
Mm-hmm, mm-hmm. Well, it's not reducing the Alzheimer's. I think it's probably accessing existent memories. So let's get into reflexes. I don't have a whole lot of time left, but I can cover what I need to here. All right, so you can classify reflexes in 4 ways, either by development, response, the complexity, sort of a morphological distinction based upon the complexity of the circuit, or exactly where it's being processed in the spine or in the brain. Yeah. So this developmental thing, is it innate or is it something that's acquired, a learned reflex with time? Response, is it a somatic reflex or is it a visceral reflex that's affecting the heart or something like that, smooth muscles of some sort or a gland? And then complexity, does this reflex involve a single synapse, or is it going to involve two or more, some sort of complex neuronal pool, polysynaptic synapse. So those are the general classifications that we have for reflexes. And we're not going to go exhaustively through them all. We're just going to look at a couple of events. So here is a basic reflex. I'm just going to put the whole thing up there. So we get some sort of stimulus. You put your hand on a tack. Ow. Sensory information comes in through the primary first-order neuron. Synapses on this second-order neuron, which is going to, the reflex is going to then synapse on this motor neuron that comes out through the ventral root and synapses on some kind of effector, a muscle, presumably a muscle that's going to pull your hand away from the stimulus, or have whatever appropriate response there needs to be to that stimulus. There's going to be a collateral off of this interneuron, this second-order neuron, which is going to ascend to where? Where's it going to go? I'll give you a hint. It's going up through the spinothalamic tract. The thalamus, right? It's going to synapse in the thalamus on the third-order neuron, and that third-order neuron is going to go into the cortex and say, oh, that hurt. I stepped on a tack. All right? So, yeah, there we go. Response by the effector. So there are these muscle spindle reflexes and tendon stretch reflexes. So we're going to hit this patellar tendon here. These two pictures are actually not related. This is for an MSR muscle. This is for a patellar tendon reflex. We stretch that. This is talking about the muscle spindle. Sorry. We stretch the patellar ligament or tendon, and it activates that muscle spindle, which then sends a signal, same way, up through the dorsal root. We have a synapse, and then we have a contraction in the quadriceps, which is relieving that tension on that transient tension on the on this tendon, transient tension on this tendon, and then this is actually inhibited. The flexors are inhibited, so that this reflex is designed to prevent, if there's too rapid an increase in tension on a tendon, is designed to prevent that tendon from snapping. So if you put rapid tension on that tendon, the reflex is designed to have a stimulation of muscles that are going to oppose that, and inhibition of muscles that would be reinforcing that. Okay, does that make sense? And so here, this is essentially the same thing. So he steps on a bottle. He gets a stimulation of the ipsilateral flexor is going to contract, and the contralateral extensor is going to contract as well. So that the leg that stepped on the glass comes up, and the other leg extends down to support your weight. All right. So we have a decussation where there's a decussation here where you get the opposite motor group on the other side having the opposite effect from that reflex as the one on the ipsilateral side. That's basically it. There's the Babinski reflex. That's not, we don't need to worry about that one. And I think that's all. Visceral reflexes happen, for example, with the heart, but let's not worry about that. So there's a recap slide, and you can go in the handout and look at it. This just is covering everything that it's an outline of everything I covered the last 5 slides. Are there any questions?